Today's topic, nitrogenase enzyme. Once again on my YouTube channel, Nurturing of Chemistry. Today I will discuss another topic of the bioinorganic chemistry, which is nitrogenase enzyme or nitrogen fixation. This is the requesting video for the student. We know that nitrogen is the essential element for the human body, because the nitrogen can form protein, amino acid, porphyrin, which is the important compound of the human body. But the major problem is that the atmospheric nitrogen cannot form this type of compound. What is the type of compound? The atmospheric nitrogen cannot form this type of compounds such as amino acid, porphyrin, protein cannot form. Atmospheric nitrogen cannot form this type of compound. Now the question arises, why atmospheric nitrogen cannot form this type of compound? From the structure of the nitrogen, we have seen that the N-N triple bond is present. Therefore, the bond dissociation energy of nitrogen is very high, and that's why nitrogen is highly inert. Now the question is arises, why atmospheric nitrogen can form the protein, amino acid, porphyrin? By the help of nitrogen fixation process, the atmospheric nitrogen can convert to the ammonia, and this ammonia can form protein, amino acid, porphyrin, which is the important compound in human life. Besides, this nitrogen is converted to ammonia by two processes. What is the process? The process is basically biological process. In biological process, nitrogen is converted to ammonia by the presence of bacteria. For example, azotobacter bacteria, rhizobium bacteria. And another process is very common process. This is the industrial method. This process is known as Haber process. In this process, nitrogen and ammonia can react with 200 atm pressure and 400 to 500 degrees centigrade. Then it can form ammonia. But this nitrogen in human body can form ammonia very easily in presence of nitrogenase enzyme. This atmospheric nitrogen can form ammonia in human body in presence of nitrogenous enzyme is known as nitrogen fixation. Now this nitrogen is converted to ammonia by the formation of two intermediate. What is the intermediate? Here this nitrogen can form the two intermediate. What is the intermediate? This intermediate is known as diazine, and this intermediate is known as hydrazine, and then it can form ammonia. And the reaction involved for the nitrogen fixation process is nitrogen plus 16 Mg/ATP plus 8H plus 8 electron. Then it can form ammonia, ADP, Pi means phosphate, and hydrogen. What types of nitrogenous enzymes are used for the conversion of nitrogen to ammonia? These enzymes are vanadium nitrogenase, iron nitrogenase, and molybdenum nitrogenase. This vanadium nitrogenase is a composed of two metalloproteins in which two metals are present. One is vanadium and another is iron. Iron nitrogenase is composed of two metalloproteins, that is What is the metal presence? Two iron center are present in case of iron nitrogenous enzyme. And molybdenum adenose enzyme, are composed of two metalloproteins and two metals are present. One is iron and another is molybdenum. For the conversion of nitrogen to ammonia, the molybdenum nitrogenase enzyme had more efficient than that of vanadium nitrogenase and iron nitrogenase enzyme. Now we will discuss the structure of the molybdenum nitrogenase enzyme and the structural activity of the molybdenum nitrogenase for the conversion of nitrogen to ammonia, and also discuss the reaction mechanism and also discuss for this mechanism reaction. Study the molybdenum nitrogenous enzyme. The molybdenum nitrogenous enzyme are composed of two metalloprotein. One is iron protein and another is molybdenum iron protein. Now we discuss the structure of iron protein. Next we discuss the structure of molybdenum iron protein. What is the structure of iron protein? This is the structure of the iron protein. This iron protein is also known as P cluster. The molar mass of the iron protein is about 60,000. In iron protein, the two Fe4S4 are linked by the cysteine ligand, the sulfur atom of the cysteine ligand. Beside, the two Fe4SO4 unit are connected by a SS bond. Here we have seen that in iron protein, one Fe4S4 unit, the iron atom is linked the sulfur atom of cysteine amino acid, and another iron atom of Fe4S4 unit are linked by sulfur atom of cysteine amino acid. Beside, with the addition of cysteine amino acid, the iron atom is also linked with the oxygen atom of serine amino acid. On the other hand, in iron protein, another Fe4S4 unit, the two iron atom are linked by the sulfur atom of cysteine residue. Now we will discuss the structure of the molybdenum iron protein and also the structural activity of the molybdenum iron protein. The molar mass of the molybdenum iron protein, that means MOFE protein, is 2,20,000 to 2,40,000. The most opposed structure of the molybdenum iron protein is shown. Here we have seen that in MOFE protein, the molybdenum atom is linked to two Fe4S4 unit by two sulfide group and MOFE bonds It is believed that the reduction of molecule of nitrogen occurs at the molybdenum site. The nitrogenous enzymes, here is molybdenum nitrogenase, in various bacteria catalyzes the activation and reduction of nitrogen, that is dinitrogen, to ammonia according to the equation. The equation is nitrogen plus 16 mgATP plus 8 H plus 8 electron, that is NH3 plus 16 mgATP plus phosphate plus hydrogens. Here the electron required for the conversion of nitrogen to ammonia. Now what is the source of the electron and how the electron transferred from P protein, that is iron protein, to iron molybdenum protein, and finally the molybdenum side, because this electron is responsible for the reduction of the nitrogen. Now the question, here the electrons are required in the reduction of nitrogen are transferred to nitrogen by the reduced form of ferredoxin. Ferredoxin means the iron protein. Reduced form of ferredoxin. The source of the electron is the oxidation of pyruvate. Now what is the source of the electron for the reduction? The source of the electron for the oxidation of pyruvate. Now these electrons are first transferred to the apiprotein. That's why the apiprotein are firstly reduced. Why reduced? Because the pyruvate supplied the electron, and these electron are going to the Fe4S4 unit of P cluster or iron protein. As a result, iron protein is reduced, and this iron protein transferred the electron to the molybdenum iron protein, and then the reducing electron to the approaches the molybdenum center, and then a series of reaction takes place. So what is a series of reaction? This reaction is molybdenum nitrogen and first lead to electron, then molybdenum, that is the diazine derivative is formed, and then the hydrazine derivative is formed, and finally ammonia and molybdenum is formed. The energy for this process is provided by the hydrolysis of ATP to ADP, and phosphate is formed. Thank you.